voices in research across the iThrive collaboration. For those of you who I've not met, my name is Karen Johnston, and I'm the director of iThrive. And just to remind everyone, the Voices series focuses on promoting dialogue and encouraging team science. The series is designed to amplify diverse perspectives in clinical and translational research, and in doing so, to foster innovation and an inclusive environment. And we at iThrive hope that all of you will use this series as an opportunity to listen more deeply to one another and to broaden your own research in the quest to improve the health of communities in Virginia and beyond. So today, it is my great pleasure to introduce my friend and colleague, Dr. Keene. Dr. Keene is Professor of Public Health Sciences and Director of the Center for Health Equity and Precision Public Health at the University of Virginia. He has nearly 20 years of experience in precision health and genomics research, with a longstanding commitment to researching with research that's focused on health disparities and health equity. His work has focused on identifying heritable risk variants contributing to complex disease. So his research program has included performing integrative omics analysis using biospecimens from underserved and marginalized populations, with hopes of alleviating the disparate burden of disease in those populations. As the inaugural director of the Center for Health Equity, Dr. Keene is working to grow the interdisciplinary center devoted to integrating precision medicine, public health, and health informatics approaches to improve the health and well-being of rural, economically challenged, and racial ethnic minority populations. Dr. Keene is a superstar in his own right. He was named as one of the top 1,000 inspiring Black scientists in America, and his impact is now international as he has become a leader in the International Stroke Genetics Consortium. Dr. Keene received his undergraduate degree at North Carolina A&T State University and his PhD in molecular medicine at Wake Forest. We are excited to hear about his journey today in his talk entitled Integrating Genomics and Public Health Approaches to Achieve Health Equity. So please join me in welcoming Dr. Keene. Keith, it's all yours.
Thank you, Dr. Johnston, for that very kind introduction. I am going to share my screen now. And give me a thumbs up if you can see the screen okay. All right, great. So what I'm hoping to do today is really give you a glimpse into my research career and who I am and how that's shaped the types of research that I've done over the years. While the topic is really focusing on integrating genomics and health equity, I'm not going to overwhelm you with science, but I really want to give you a kind of a snapshot of the types of research that I've done over the years, why it's important to me, and what this type of research will look like. So again, thanks for having me and thanks for joining for this talk. So I want to start first by doing some acknowledgements. As a human genetics researcher that's kind of moving into the space of health equity, none of this work would be possible without the large interdisciplinary teams, which include individuals across multiple institutions, across different disciplines and various expertise, including genetics, clinical care, community engagement, biostatistics, et cetera. But most importantly, I think none of this work would be possible without not only the funders, but also the participants and the study staffs that allow us to recruit individuals that are part of our studies, as well as those citizen or community advisory boards that really help shape our study designs and how we approach and disseminate our findings. So just to elaborate a little bit more about me and to give you more context as to why this line of work is very near and dear to my heart, I grew up in a very rural area in eastern North Carolina. County population is less than 20,000 individuals. The town that I grew up in, there were 800 or less of us. So growing up as a kid, I had no idea what a research scientist did. much less what genetics research looked like. As a matter of fact, my aspirations entering college, as most undergraduate biology students, was to attend medical school. But all of that really changed for me the summer after my sophomore year, where I was able to do a summer research internship as a Howard Hughes Fellow at the University of Iowa, studying genetics in Arabidopsis, looking at the genes that contribute to delayed senescence. I was also fortunate enough that next summer to conduct another research internship at North Carolina State, again, looking at genetics in plants, this time looking at Metacargo truncatula to identify nematode-resistant genes. But most of that really changed for me during a graduate career fair where one of the representatives was the Wake Forest School of Medicine, and a program in particular that really stood out to me was the molecular medicine PhD program. And the unique thing about this program is, while its focus was on bench to bedside, so translational research, the students were given the opportunity to do clinical rotations after the first year of study. So that kind of gave students a sense of not only the clinical applications of the research, but also how it could be very impactful. So to me, that was the selling point. And once I did my rotations, one of the labs that I rotated in was the laboratory of Dr. Michelle Sell, who was doing genetics research in type 2 diabetes and end-stage renal disease with a focus on African-American populations. So it was really a no-brainer for me. And I joined the lab, and I would say those were some of the best years of my life doing that type of research. But also, this is just I just want to make a plug here for any students, whether you are undergrad thinking about PhD programs, whether you're already in a PhD program, or whether you're thinking about a postdoc. Be very intentional about selecting the mentor that you choose, and which lab you rotate in, or you stay to do your degree or do your postdoctoral training. I would argue that this was the single most important decision of my research career, and really paved the way for my career trajectory. So I owe a debt of gratitude to Dr. Sell for all of the mentoring over the years. Also, this is a note to PhD mentors and advisors. Please do your best at mentoring, because it really can make a huge difference in the careers of these trainees. So the research approach we use as a PhD student, at least early on, was to identify our favorite gene, whether it was a candidate gene based on biology or biochemical pathways, or if it was a gene identified through family linkage studies. So with the case of type 2 diabetes, you can imagine that glucose transporters or genes involved in the insulin pathway would have been ideal candidate genes. But then we would then take those genes and do what we call fine mapping to really catalog all of the variants across those gene regions, and ultimately try to determine the genetic variant that was most significant and likely to be determined as the causal variant contributing to the association for that phenotype. In 2007, after graduating from Wake Forest, I decided to pursue my postdoctoral studies at UVA in the Center for Public Health Genomics. This was a newly established center that was really at the cutting edge of genomics and public health research, with an emphasis on type 1 diabetes. I joined the laboratory of Dr. Pat Concannon, who was a type 1 diabetes researcher, and it allowed me to shift my studies away from the traditional case control cohort to look at type 1 diabetes, which again is a disease that is not very common in African American populations. But it allowed me to do studies in family-based and learn additional types of analyses. So I would say my early years really focused on the genetics of type 1 and type 2 diabetes, and we were very successful during that time with a number of publications ranging from candidate genes in the ENPP1 and ESR1 genes to exploring type 1 diabetes associations and trying to figure out how to account for population substructure and admixture in our genetic studies of African individuals, African ancestry individuals. But the field of genomics research really changed around 2003 with the completion of the Human Genome Project. And this really set the stage as serving as a reference population for all genetics studies, creating this reference genome that was completed again in April of 2003. Now, as we are more than 20 years removed from the completion of this project, this has single-handedly transformed how we think about genomics research, has transformed my research career, and how we've been able to impact the field of not only precision health, but also integrative and multi-omics. Additional advances, such as improvements in genome sequencing technologies, have also changed the way we do research. So during the early days of the Human Genome Project, where it costs hundreds of millions of dollars to sequence an entire genome, you can see over this 20 to 25-year stretch, now we are at approximately the $1,000 genome, where you could literally sequence the entire genome for less than $1,000. So again, that has shifted not only how we think about and design our genetic studies, but instead of going from traditional candidate gene studies, now we're able to transition to what we call genome-wide association studies, or GWAS. And GWAS really allow us to interrogate genetic variants, usually in the form of single nucleotide polymorphisms, or SNPs, which is demonstrated by this G to T change here. But it also allows us to evaluate the entire genome for associations with various diseases and phenotypes. The beautiful thing about genome-wide association studies is that it requires no prior knowledge of the gene or variant function in relation to the disease or of interest. So essentially, this is a gene hypothesis generating type of approach. So for those of you not familiar with GWAS, I'll just walk you through it really briefly so you can get a full understanding of why community engagement and diversity is important, and when I bring that up in some of my subsequent slides. But we generally recruit a large population of individuals with and without a disease. So let's just assume here in this case, we're talking about type 2 diabetes. So this large group will be individuals with the disease, and this group will be our controls, or individuals without the disease. We then can sequence or genotype hundreds of thousands to millions of variants across the entire genome, perform subsequent statistical analyses to identify variants with different frequencies in our disease versus our non-disease populations. And this middle graph here is what we call a Manhattan plot. And this is used to illustrate the p-value, the association results of GWAS, where on our x-axis we have our chromosomes here, labeled 1 through 22. And then on the y-axis, we have our negative log p-value. Each one of these points, these colored points, corresponds to a p-value for a given variant. And this dotted line here usually represents our genome-wide significant thresholds, accounting for multiple testing. So any dots you see above this line represent what we would call genome-wide associations. And once we've identified those statistically significant associations, the next step is usually to identify or replicate those in independent populations. So for those of you that are familiar with companies such as 23andMe, many of the variants that they include in their disease panels are generated through studies such as this, where then this is a clear example of how our research can inform commercial products. So over the years, I would say over the last 15 or 20 years, genome-wide association studies have been extremely successful. Dating back to February of 2022, there have been thousands of publications, hundreds of thousands of variant trait associations that have really provided insight into the mechanisms of various diseases, ranging from diabetes to cancer and others. But GWAS have not been perfect. while they've identified a large number of associations and have advanced some of the statistical methods, allowing us to generate what we call polygenic risk scores, there have been struggles with replication across multiple populations, as well as issues with identifying the true causal or actionable genes. So these are genes or alleles that may have clinical utility, or where their function or mechanism of action is directly correlated with a particular pathway. So this is what we refer to as missing heritability. And GWAS have not explained all of the expected heritability for these different diseases with genetic influences. So some of this is based on the design of genome-wide association studies. Again, they're traditionally designed to capture common variations. So we are not able to account for things such as rare variants or structural copy number variants, et cetera. But one of the true shortcomings in the field of genomics is the lack of inclusion of diverse populations. And I always like to refer to this Nature publication from 2016, where the authors did a catalog of all of the genome-wide association studies in 2009, and then they reassessed in 2016. And what they found is that while the percentage of individuals of European ancestry had decreased between those two time points, the number of individuals of African ancestry, Latin American ancestry, Hispanic individuals, and indigenous populations had relatively remained unchanged during that time period. So what we saw was that the major differences was actually in the increased number of individuals of Asian ancestry contributing to these genome-wide association studies. So one may ask, why is this so important? And this is one clear example from a publication from 2016 that really demonstrated that a lack of diversity in these genetic studies was leading to misdiagnoses for this heart disease. And what they found is that the genetic backgrounds in African Americans was very different than the individuals that were included in the studies that led to the diagnostic tools. So this was really creating an issue in the field of cardiac genetics. Another major region is we are in the process of a major change in our demographics across our country. It is projected that by the year 2050, people of color will make up more than half of the US population. So one might argue if individuals of diverse ancestries are not included in our genetic studies, that the findings from genetic studies will not be applicable to a large majority of the population in less than 30 years. So again, to bring this all full circle and how this kind of shapes or has shaped my career, around the year 2010, as we were transitioning into the GWAS era in genomic study, I was in the process of transitioning from postdoc to trying to figure out the next chapter of my career. So ultimately, I decided to accept a position as a research assistant professor to stay at UVA to work with a group that was looking into pharmacogenetics and stroke genetics research. So this is one of the major pivots in my career, and where we were now trying to use genetics and pharmacogenomics as a way to address precision or personalized medicine. And for those of you unfamiliar with the concept of precision or personalized medicine, this usually applies to drug and treatment response. And the goal of precision medicine is to truly eliminate the one-size-fits-all approach that is used for drug prescriptions and usage. So in this example here, let's say we have this large population. Let's assume this entire patient group are all type 2 diabetes individuals that have the same diagnosis and they're given the same prescription. The concept of precision medicine says that we want to identify individuals that will have no benefit and therefore should not be prescribed. So the first step is obviously these individuals in the red, where the drug is toxic and not beneficial, these individuals should never be receiving this drug. Likewise, for this individual group here that was in the yellow, where the drug is not toxic, but it's also not beneficial, they stand to gain nothing from being prescribed this medication. So in reality, we're trying to identify the right populations that are stand to have the most benefit from these drug prescriptions. So in this case, the individual group here, circled here, where the drug is not toxic and also it is beneficial, they stand to gain the most. Whereas this group in green, where the drug is slightly toxic, but there are benefits, those individuals might benefit from dosing adjustments in this case. So again, the goal of precision medicine is really to give the right treatments at the right time, every time, to the right person. So I was able to step into the world of pharmacogenomics using this study population from a clinical trial called the Vitamin Intervention for Stroke Prevention. And this was a clinical trial that was designed to see if stroke patients who received B vitamin supplementation would have a reduced rate of recurrent stroke, myocardial infarction, or mortality. And one of the nice things about this is that it included a diverse population, the multi-ethnic population, which although the numbers are a little small here, but it allowed us to think about treatment response, not only from the clinical trial perspective, but from differences across race, ethnicities. And in the next couple of slides, I'm really going to highlight some of the disparities that we see in stroke. But again, this opportunity to look at treatment response in a clinical trial that was well phenotyped really set the stage for my independent career. And again, I was fortunate to work with my former PhD advisor, as well as a very enthusiastic neurologist at UVA, Dr. Brad Worrell. So stroke, every 40 seconds, someone in the United States suffers a stroke. Unfortunately, every 4 minutes, someone is going to die from a stroke. Stroke is the 5th leading cause of death in the United States and the #1 cause of serious long-term disability. For individuals that are fortunate enough to survive an initial stroke, 40% will have a recurrent stroke within 5 years of that first stroke. So we know from maps like this from the CDC that stroke prevalence, incidence, and mortality rates differ drastically across the United States. So for those of you that have seen these before, you know that these dark purple areas represent higher mortality rates and typically cluster in the Southeast United States, a region we commonly refer to as the stroke belt. But in addition to differences across regions and geographic spaces, we know that different race ethnic groups have different, are impacted differently by stroke. In particular, African Americans are the most impacted, have the highest disparities for stroke. African Americans in general are more impacted by stroke than any other racial group. They're more than 2 times likely to have a first stroke. They typically suffer their strokes earlier in life, and those strokes are usually more severe or disabling or more likely to cause death. As I told you in my intro, I was born and raised in Eastern North Carolina, but also my previous faculty position was at a university in Eastern North Carolina. So some of our task was really to think about how we can address disparities. And what you can see in this table here is that Eastern North Carolina as a whole has higher mortality rates independent of race ethnicity across all groups compared to not only the state average, but also the national average. So this is a topic that was very near and dear to me personally, as this affects the region that I was born and raised at significantly higher rates than the rest of the nation. But from a research perspective, I was really honored to be a part of the Consortium of Minority Population Genome-Wide Association Studies of Stroke. I know that's a mouthful, but we refer to that as COMPASS. And I was the co-lead of this initiative for several years. And through this initiative, we were really focused on creating this coalition of individuals that not only had genetic data for individuals of African ancestry, but also had stroke and phenotype data for those. So through that initiative, we were able to contribute to some very important multi-ancestry GWAS studies for stroke, which have identified more than 32 loci associated with stroke and stroke phenotypes. And this was in nearly 520,000 individuals. But what was more important for me was the opportunity to lead the CHEPPH effort to generate the first two large-scale GWAS studies of African ancestry individuals with stroke. And I'm happy to say that this is our second effort here, which was published in 2020, but it really was the largest GWAS of stroke in African ancestry populations to date. And although we say the largest relative to the initial study I presented with the 520,000 individuals, we were still only able to identify less than 4,000 stroke cases and 18,000 stroke controls. Again, this is coming from a population that is significantly impacted by stroke, and yet we still have these relatively small numbers, which further supports that idea that genomics is failing in terms of diversity. This project was actually awarded the 2020 Stroke Progress and Innovative Award third place prize from the American Heart Association. So this again was one of my prized accomplishments throughout my research career. But what we learned is we are really good at identifying the genetics involved in various diseases. Although we've only been able to explain a certain percentage of that heritability, we recognize that there are other forms of omics or other data types that may be contributing to the overall these overall complex diseases. So we decided to expand our approaches, again, encompassing this research or team science approach, and really spanning out to look at integrative omics to capture epigenetics or epigenomics, metabolomics, among others. So for those of you that are not familiar with epigenetics, I'll just briefly introduce it to you. So epigenetics refers to chemical alterations in DNA and or associated histone proteins that influences the genome without altering the genetic sequence. So we're not changing that ATGC sequence. We're really modifications, whether it's in the form of DNA methylation or histone acetylation, et cetera, that influence the accessibility of DNA and its ability to go from DNA to RNA to protein. We know that epigenetic factors can be both stable and dynamic. They can be influenced by environment and behaviors, whether it's diet and exercise or other things. We also know that epigenetics is important in different biological processes and diseases, most notably cancer. But there's also a growing literature that suggests that epigenetic modifications can be passed down across generations. So this has the potential to explain some of the effects of trauma or early life experiences that can be passed down from one generation to the next. And I always like to show this model because visually I think this drives home the point where we have essentially two identical, from a genetic perspective, animals where they differ only in the methylation status at a particular locus, which causes one animal to be this yellow goldish color and obese, and the other animal to be more the natural brown color and normal size. So this visually just shows that epigenetics can significantly influence phenotype and other traits. So we were able to expand this data set. As I mentioned earlier, we were able to genotype all 2,100 VIS participants with a GWAS array, but we've also been able to recently expand those to look at a subset of DNA methylation loci, as well as a large panel metabolomics. So again, I'm not going to go into all of the details, but just suffice to say that with the methylation or epigenetic studies, we're able to discern DNA methylated sites from unmethylated sites to kind of generate this score that indicates methylation state. So we've done a number of analyses ranging from genetic studies in stroke and stroke recurrence. We've done a number of epigenetic wide association analyses, identifying several loci in both our race ethnic stratified, whether it's African American only or European American ancestry. And these have resulted in a number of publications from different journals over the years, as well as a number of funding mechanisms or proposals. But what we've realized is, as the field is starting to evolve, where the NIH is now requiring consideration of sex as a biological variable, the inclusion of diverse populations, that we needed to change our approach a little bit to kind of keep up with the changing times. So traditionally, we've thought about health disparities in the context of just the disproportionate burden or differences of disease or death by a number of categories, whether it's race, ethnicity, sex, gender, age, geographic region, etc. And we typically think of this as incidence rates, prevalence rates, or mortality rates, similar to what I showed you in the CDC maps. But lately, the shift has been moving more towards this concept of health equity, which really centers on the idea that every person has the opportunity to attain his or her full health potential, and no one is disadvantaged from achieving this potential because of their socially determined circumstances, which we often refer to as social determinants of health or social drivers of health. And these drivers can usually be grouped into five kind of domains. One is your education access and quality. The other includes healthcare access and quality, neighborhoods and built environment, socioeconomic status, and social and community context. So again, as a stroke researcher, you can imagine that access to care may be a critical factor in stroke survivorship. If you live 60 miles from the nearest hospital and you suffer a stroke, survival may be significantly impacted. So typically we think of health disparities as the tip of the iceberg, but when we think of health equity, we really must capture this entire picture to get at one's overall risk profile or disease risk profile. And one thing that we haven't done a really good job of is taking into account both historical and childhood trauma, as supported by the epigenetic slide I mentioned a few minutes ago. So we've really started to transform how we do our research and how we design our studies to think beyond the traditional GWAS and EWAS to factor in social and structural determinants of health, including all of these that I mentioned a second ago, and how they can influence not only the risk factors for stroke, but also how they can be mediated by the environment and environmental exposures. And I'm happy to say that we have an R1 that is pending funding. So we're hoping for the notice of grant award any second now to really explore the contributing factors of social and structural determinants of health in regards to stroke and stroke recurrence. So while I've highlighted most of my personal research interests, since 2018, I've really had to think to kind of change the direction in terms of how I think about not only research, but also the overall research enterprise. So in 2018, I was asked to serve as the interim director for Center for Health Disparities at my previous institution after the sudden retirement of my boss. And the overall goal of that center was really to think about how we can reduce disparities and promote health equity through not only traditional research, which I've spent the bulk of this time talking about, but also other initiatives that encompass community outreach and engagement, as well as education and training pipelines. So this is another one of those major transition or pivot points for me, where I've really had to start thinking beyond just Keith's research lab in stroke genetics, but thinking bigger picture. And part of that bigger picture is really thinking about this new field of precision public health, which is centered at delivering the right interventions to the right population. And we saw a little bit of this during the COVID-19 pandemic with the incorporation of big data and technology to do community surveillance and targeted interventions, whether it's thinking about vaccine hesitancy or things of that nature. So this is something that's truly gained a little bit of traction since the start of the pandemic. But again, to address all of the big pictures, and as a center director, we have to think bigger than just our individual labs. This just kind of gives you a snapshot of some of the themes or some of the areas that we think about in these health disparities, or in this case of UVA, health equity centers, where our research can range from anything as basic, from basic sciences and genetics all the way to public health and epidemiology, where we can think about community engagement, not only in terms of health education and health literacy, but also in some of the services we can provide through health fairs, efforts to increase recruitment in biomedical research and clinical trials, engagement in community advisory councils or community advisory boards, and then thinking about ultimately how we can train and prepare the next generation of the workforce, whether it's through earlier exposure to youth as far as education opportunities or career options, whether it's undergraduate research, internships, workforce development, et cetera. And I'm going to give a few examples of some of the things I've done over the past couple of years. But I really want to take this time here to kind of introduce CHEPPH a little bit, and how there's an opportunity for us to think about how to work together in the broader sense to address some of the goals of not only iThrive and CHEPPH and our respective institutions, but how we can make a bigger impact across the region. So as Dr. Johnston mentioned in her introduction, CHEPPH, or the Center for Health Equity and Precision Public Health, is an interdisciplinary center that's devoted to integrating precision medicine, public health, and health informatics to really improve the health and well-being of rural, economically challenged, and racial ethnic minorities. So again, this brings together all of the concepts of not only public health and genomics, but also keeping in that health equity lens throughout the entire process. So the overall goal is really to utilize inclusive approaches to reduce disparities and ultimately promote and achieve health equity. And again, this can only be done by research, as well as community outreach and engagement, and education and training pipelines. So while CHEPPH is centered in the School of Medicine and funded primarily through the Center for Public Health Genomics and the Department of Public Health Sciences, it truly is supposed to be a bridge across not only the university, but across multiple entities, which can include UVA Health, our healthcare system, and a number of industry partners, community partners, and partnering universities. So I consider myself a friendly place to collaborate for anyone on the call that finds any of this interesting and exciting. So the research mission of CHEPPH is pretty simple. We want to figure out how to create an integrative research agenda that helps better understand the biological, the social and behavior, and the physical and environmental exposures that contribute to health outcomes. So what does this look like from my perspective? When I think of biology, I'm always focused on the multi-omics. How does your genome, how does your epigenome, how do those things contribute to disease risk? But also when we think about social and behavior, we can think about tobacco use, alcohol use, physical activity, the foods we eat, et cetera. And then clinically, I think this really represents a new area that we should be thinking about ways to expand. How can we tackle some of that electronic health data to really address either disparities or inequities, or figure out ways to either target certain populations or improve care, overall health care? And then again, keeping in mind that the environment in which we live is very critical to our overall health and well-being. So I just want to give a couple of examples of things that I've done over the years, and I know we want to make sure that there's plenty of time for question and answer, so I'm going to wrap up here shortly. But this is just one example of a Health Equity Roundtable that I sponsored with funding from Bristol Myers Squibb, with collaborations from not only community health coalitions, but also various institutions across the Mid-Atlantic and the Southeast, as well as state public health officials. And this was literally about 2 years ago. On the education and training front, I really try to make an emphasis to diversify the workforce, the future workforce. So one example is funding that we received from the North Carolina GlaxoSmithKline Foundation. This was a $1.4 million grant that was awarded as a partnership between my former institution and a historically Black college in North Carolina, Fayetteville State University, where we were really trying to enhance student success and diversify the public health workforce by creating a summer research program, as well as incorporating a scholarship program into the MPH program at my former institution. I'm happy to say that I was also a multiple PI on a NIH Mark T34. This is focused on getting undergraduates research exposure as well. So I hope it's been clear throughout these last 30 or 35 minutes that diversity and inclusion is essential to success of our research enterprise and to addressing health equity. And I always like to include this slide, one, because it sounds good with the rhyming, but also because I think it really drives home the message. So the first point is representation matters. When you have people who look and sound like the communities they serve, we generally have better outcomes. And I think data is really showing us, especially in the clinical space, that minority patients have better outcomes when they receive their care from individuals of their own race and ethnicity. So representation can then lead to improved participation, where we can increase the diversity in research and clinical trial studies. And then as a result of that diverse participation, that can lead to better, more generalizable translation, where we are generating data and results that are relevant and applicable to a large number of diverse populations, especially given the changing in demographics, that slide I showed earlier. And then ultimately, that improved translation can result in elimination, where we can cure disease or eliminate disparities or other barriers. So again, representation leads to greater participation, better translation, and ultimately elimination. So we know where we stand as a state. We know what the major killers are, what the major causes of death are, and this just highlights some of those. So we know the usual players, the heart disease, the cancers, stroke, etc. We also know the unique features of our state. We know that individuals that live in rural areas often have poor health outcomes than individuals that live in urban areas. And clearly this map demonstrates that we have a significant amount of rural areas across our state. And even studies have shown that, again, there are differences in health outcomes. This example is for heart failure, but the same could be true for a number of diseases. We also know that money matters. Across the state, we have several large pockets of poverty, and poverty influences health outcomes. And we also know education matters. While there's probably a strong correlation between education and income, we also know that health literacy is a major factor when it comes to understanding not only your health risk, but what types of medications and adherence and things like that. So I want to close with this slide here again, just to drive home the point that if we want to address the issue of health equity, we must include diverse teams with various expertise, and we must address all of these causes to really get at what the underlying factors are that are creating these disparities and these inequities. With that said, Karen, I think I'm right at 40 minutes. And I hope I have left plenty of time for us to have conversation. So I'm going to stop sharing, and I'm going to turn the floor back over to Dr. Johnston.
Awesome. I just love hearing you talk, Keith, because of all the components